Hello students. Today, before starting my lecture, I am going to show you something. A potted plant was covered with a transparent plastic. If we put this plant in a bright sunlight, we will see that the packet was hazy. After some time, water droplets start accumulating in the inner side of the plastic bag. Now, some question may arise. First, why the plastic become hazy? Second, why water droplets starts accumulating? And where does this water coming from? We will get this answer today in the topic Transpiration, Significance, Factors affecting transpiration, mechanism and its measurement. So, in this topic we will discuss on transpiration and try to know the different kinds of transpiration. We will try to understand the significance of transpiration. We will also study the factors affecting the transpiration. We will know about the stomata and the mechanism that regulates its opening and closing. Finally, we see the different processes of measurement of the rate of transpiration. Now, transpiration. It is a physiological process. It can be defined as loss of water in the form of water vapor from the aerial parts of the plant. This is just to keep water balance within the plant that they draw from the soil. For drawing water, they have to transpire. Now, depending on the sites or regions from where the transpiration takes place, transpiration are divided into three types: stomatal transpiration, cuticular transpiration and lenticular transpiration. First, I will want to show you or explain you what the lenticular transpiration means. Lenticel. This is an area in the bark where loosely arranged cells are present. They are called complementary cells. Water gets evaporated from these loosely arranged cells. This type of loss of water is termed as lenticular transpiration. Cuticular transpiration. Cuticle is a waxy layer that is present on the epidermal surface. Though cuticle is present to prevent transpiration, but sometimes from the cracks and thin layers of cuticle, water evaporates. This is known as cuticular transpiration. Very less amount of water get transpired from lenticels as well as from cuticle. The most water get evaporated from the stomatal pores. Stomata are the fine pores present on the epidermis of the plant, mainly on the leaves and in some stem. So, loss of water from this stomata is known as stomatal transpiration. Now, I will want to explain you the advantages and the disadvantages of stomatal transpiration, or say transpiration. It is better I would read out the advantages first. Transpiration creates succession force and help in the ascent of sap. It affects the diffusion pressure deficit and promote diffusion through the cells. It affects absorption of water and minerals by roots. It helps in evaporating excess amount of water. It also plays a role in translocation of food to different parts of the plants. It maintains a suitable temperature for the leaves. It brings about the opening and closing of stomata, which is indirectly influence the process of photosynthesis and respiration. So, looking at these advantages, transpiration is beneficial for plants. Now, regarding disadvantages, transpiration consumes energy and cause unnecessary absorption of excess water by roots. Secondly, when the rate of transpiration is high and the soil is deficient of water, the plants get wilted. Transpiration is the cause of death of millions of plants every year. So, considering the advantage and the disadvantage, Curtis depicted transpiration as a necessary evil. Now, I will discuss the different factors that affect rate of transpiration. The different factors can be divided into two heads. One is the external factor and another is the internal factor. Firstly, let us consider the external factors. Temperature. Temperature has a direct relation with rate of transpiration. More the temperature, more is the rate of transpiration, because transpiration removes or lowers the relative humidity and help in opening the stomata widely. Second is atmospheric humidity. Humidity has inverse relation with transpiration. When the relative humidity is high, the rate of transpiration is low. This is because when the atmosphere is saturated with water or water vapor, the diffusion of water vapor from the leaf is restricted. So, more the humidity, less is the transpiration rate. Wind velocity. When the wind is static or stagnant, the rate of transpiration is normal. But blowing of wind gently increase the rate of transpiration, because flow of air removes humid air from the vicinity of the plant. So, more and more water vapor will diffuse into the atmosphere. But a violent wind reduces or stops transpiration, because violent wind leads to closure of stomata. Light. Light increases transpiration because light promotes stomata to open and it also increases the temperature. Carbon dioxide. Carbon dioxide has an inhibitory action on transpiration. An increase in concentration of carbon dioxide in the atmosphere over the usual concentration inside the leaf retards transpiration because it leads to closure of stomata. Available soil water. Rate of transpiration will decrease if there is not enough water in the soil. Antitransparent. Chemical substances which reduce the rate of transpiration is known as antitransparent. Some of the antitransparent chemicals are phenyl mercuric acetate, aspirin, abscisic acid, and even low viscous wax. Now, the internal factors. Among the internal factors, first is the stomatal frequency. Stomatal frequency is the number of stomata per unit area of leaf surface. So, more the frequency, and if all the stomata are open, then rate of transpiration will be more. Structural peculiarities. Certain plants are adapted to reduce the rate of transpiration by reducing the size of the leaf, forming sunken stomata, synthesizing thick cuticle, etc. Other factors. Several other factors such as leaf area, amount of spongy tissue, volume of intercellular spaces, orientation of leaf and extent of root system also affect the rate of transpiration. Now, to know the mechanism of transpiration, we have to understand the structure of stomata, as we have studied that opening and closing of stomata regulate transpiration. Stomata are very minute opening or pore on the epidermal layer of all plant organs except the root. The pore is surrounded by two guard cells. These guard cells are responsible for regulating the opening and closing of stomata. The guard cells are either kidney-shaped or dumbbell-shaped. The inner wall of the guard cell is thick as compared to the outer wall. Each pair is joined at the ends. The cell wall of the guard cells possesses cellulosic microfibrils. These cellulosic microfibrils are oriented around the circumference of the elongated guard cell in a radiating manner from the center of the stomata. This microfibrillar arrangement is known as radial methylation. Due to the radial methylation, when a guard cell swells up, absorbing water from the surrounding cell, it cannot increase in diameter. Instead, it increases in length. As a result, the pore of the stomata get wider and we call the stomata is open. Now, considering variable behavior of stomatal movement, 5 categories have been recognized. Photoactive movement. Some stomata, they are sensitive to light. They open during day and closes during night. Scotoactive movement. It is just the reverse of photoactive movement. Here, the stomata open at night and closes during day. Hydroactive movement. In certain cases, stomata close due to excessive loss of water from the epidermal cells and open due to turgid condition of epidermal cells. This is usually found during midday. Autonomous movement. In some cases, stomata show a rhythmic movement, that is, they open for a 15 or 20 minutes and after that they closes up and again they open after an interval of time. Passive and active movement. Opening of stomata is considered as active process while closing is the passive process, and this is caused by the turgor change in the guard cell. Mechanism of opening and closing of stomata. The opening and closing of stomata depends upon the change in the turgidity of their guard cells. Different theories have been proposed by different scientists to explain the changes in the turgidity of the guard cells. First, theory of photosynthesis in guard cell. Von Moll in 1956 observed that stomata opens in light and closes during night. So he proposed that chloroplast in the guard cell produce carbohydrate in the presence of light by photosynthesis. The photosynthates, being osmotically active, draw water into the guard cell from the surrounding cell. As a result, the guard cell becomes turgid and the stomata opens up. At night, since photosynthesis is not occurring, so no photosynthates are produced, so cells get flaccid as it fail to draw water from the surrounding cells. Instead, the water removes from the guard cell and enters into surrounding cell. As a result, the stomata closes. But the theory has lots of objections. First, starch has been found in very young guard cell even before it has emerged from the bud. Starch has also been observed in guard cells of leaves kept in dark and also in albino leaves. In camp plants, stomata open during dark or during night. Chloroplast of monocot guard cells are non-functional, that is inactive photosynthetically. Thus, synthesis of starch in the guard cell is not dependent on photosynthesis. So, this theory does not get any preference. Second theory is theory of starch-sugar interconversion. Sayre in 1926 noted that stomata are sensitive to pH. At high pH, that is pH 7, stomata opens, while low pH favors closing of stomata. In and Tang in 1948 obtained evidence of the presence of a phosphorylase enzyme that is pH sensitive. They observed that at pH 7, the enzyme phosphorylase transforms starch into glucose, while at pH 4, the same enzyme converts glucose into starch. Thus, the mechanism of opening and closing of stomata can be explained as: in the morning, due to photosynthesis, carbon dioxide concentration lowers in the mesophyll cell and guard cell. As a result, pH of the guard cell rises. It stimulates enzyme phosphorylase. Phosphorylase converts starch into glucose 1-phosphate. The latter is changed to glucose 6-phosphate, which undergo hydrolysis to produce active glucose and phosphoric acid. Evening closure of stomata is brought about by increased carbon dioxide content in the guard cell and in the mesophyll cell, because photosynthesis is inactive at that time. As a result, respirative carbon dioxide get accumulated in this region. The decrease in pH of the guard cell brings about phosphorylation of glucose. In the presence of phosphorylase, glucose 1-phosphate is changed into starch. As a result, osmotic concentration of the guard cell falls. They lose water to adjacent epidermal cells. With the loss of turgidity, the guard cell shrink and closes the pore in between them. This theory has also some objections. They are: glucose is not found in the cells at the time of stomatal opening. Starch-sugar change are chemically slow, while opening and closing of stomata are quite rapid. Wide change in pH of guard cell cannot be explained on the basis of carbon dioxide concentration. Blue light has been found to be more effective than other wavelength of light during opening of stomata. The same cannot be explained by starch hydrolysis theory. Hydrolysis of starch cannot account for high rise in osmotic pressure found in the guard cell. Finally comes the theory of proton transport and hormonal regulation. Levitt in 1973-74 suggested that the turgidity of the guard cell of many species of plant is regulated by potassium ion, chlorine ion, hydrogen ion and malate. Now, when plants are exposed to light, the guard cell accumulate large amount of potassium. The potassium accumulation is due to the operation of an active exchange process. The ATP from respiration required in the ion exchange process and may come through respiration. Protons are pumped out of the guard cell. Instead, potassium ions are influxed into the guard cell. They also promote or allow chlorine to enter into the guard cell. Inside the guard cell, these potassium, they combine with malate to form potassium malate or dipotassium malate. This potassium malate, dipotassium malate as well as chlorine, they are highly osmotically active compounds. They promote inward flow of water into the guard cell from the surrounding cell. As a result, the guard cell gets turgid and stomata opens up. Closure mechanism involves participation of inhibitor hormone ABA or abscisic acid, which functions in presence of carbon dioxide as well as during stress. In the presence of ABA, the permeability of the membrane changes. As a result, the potassium ion, they go out from the guard cell and influx of hydrogen ion takes place. Removal of potassium ion from the guard cell also take away water. As a result, the guard cell becomes flaccid and stomata closes. There are some roles of carbon dioxide also in stomatal movement, which I have communicated during my lecture. It has been observed that if a leaf is placed in a stream of air free of carbon dioxide, the stomata will open even in absence of light. But if the carbon dioxide concentration is above atmospheric level, that is about 330 parts per million, then it was observed that the stomata closes even during day. The reason behind closing of stomata is that carbon dioxide dissipates the proton gradient across protoplasmic membrane within the guard cell. When the proton gradient is dissipated, active transport of potassium ion into the guard cell ceases. Guard cell disposes of accumulated ions, potassium ions is transported out of the guard cell, and this leads to removal of water from the guard cell becomes flaccid and stomata closes. In CAM plants growing under condition of water stress, malic acid decarboxylation occurs through the day and internal tissue accumulates carbon dioxide. As a result, carbon dioxide concentration within the cell increases during day. This leads to closure of stomata during day. At night, these carbon dioxide, they get fixed into malate, and as a result, the removal of carbon dioxide increases the pH level and that allow the inflow of water within the guard cell. As a result, the guard cell becomes turgid and stomata opens at night. Blue light also play an important role in transpiration. Zeaxanthin is the blue light receptor present in the light harvesting complex 1 and 2. In presence of blue light, zeaxanthin get excited. Excited zeaxanthin cause conformational change in an apoprotein. Calcium ATPase in the chloroplast membrane due to this get activated. It take up calcium in the chloroplast. Low cytosolic calcium ion activates hydrogen ATPase in the cell membrane. Efflux of hydrogen and influx of potassium takes place. Influx of potassium also promotes chlorine inside the cell. As a result, the cascade of inflow of water takes place and as a result, stomata opens up. So, these are the theories which depicts the transpiration. Now, how we can measure the rate of transpiration. Measurement can be done by weighing method and by an instrument called potometer. In case of weighing method, either a potted plant whose lower part is covered with polythene can be initially weighed and kept in the sunlight for some time. After some time, the pot is reweighed and the loss of water is indicating the rate of transpiration. Similarly, taking a potted plant in a closed container and connecting it with a tube containing calcium chloride, where the calcium chloride is dry, it has the ability to draw water into it. Weighing the dry calcium chloride initially and weighing it after some time indicate the change in weight in the calcium chloride, and this change in weight due to the amount of water released by the plant. This is the second weighing method through which rate of transpiration can be measured. Potometer is a device where one arm is fitted with a plant. It contains a funnel where water is poured, and the other arm of the instrument is connected with a capillary tube containing a measuring scale. Inserting a bubble in the capillary and measuring its initial position, putting the entire instrument for few hours, we observe that the bubble will move from its initial position to some other position. Change in the position of the bubble indicate the rate of transpiration during this period. So, in nutshell, we observed that loss of water in the form of water vapor from the aerial part of the plant is known as transpiration. Transpiration is beneficial to plant because it helps in drawing water due to transpirational pull. It promotes water balance within the plant. It leads to transport of minerals from root to different parts of the plants. But on the other hand, excessive loss of water may lead to death of the plant. So, transpiration can be considered as a necessary evil. We have observed different theories which try to predict the ways through which water enters into the guard cell, which leads to opening of the stomata. And this opening and closing of stomata is basically related with transpiration. Finally, we observed the methods through which we can determine the rate of transpiration. Thank you.